Good morning. Episode 3, Why Acidic Mobile Phase A is Used in Pharmaceutical Analysis. Typically, acidic mobile phase at pH 2 to 4, such as TFA, trifluoroacetic acid, formic acid, or formate buffers, are typically used in pharmaceutical analysis. Reason number one, because 70 and 80% of small molecule drugs are weak bases, which are protonated at low pH. However, even when they are ionic and protonated, there are sufficient hydrophobicity in the rest of the molecule for adequate retention in reverse phase chromatography. Second, acidic mobile phase A at pH 2 to 4 is desirable because it suppresses ionization of residual cyanol group, which causes excessive peak tailing of basic drug due to acid-base ionic interaction. Let's explore that a little bit later. Here is a schematic of silica and bonded phases courtesy of Waters Corporation. Note that bonding and end capping from these bonded phases provide incomplete coverage since most of the cyanodes are inside the pore. So there's always residual cyanide group left. And it's problematic because they are acidic. Residual cyanide are acidic and can cause peak tailing of basic analytes. The reason why acidic mobile phase at pH 2 to 4 is desirable because above 4, the SiOH group can actually be ionized into SiO-1, which is become ionic, which have much stronger acid-base ionic interaction, causing excessive peak tailing for basic drugs. Prior to 1980, there are really two big problems in preventing reverse phase chromatography in drug analysis. First one is peak tailing for basic drugs. And second is even bigger, which is the batch-to-batch reproducibility problem. So prior to 1980, to reduce peak tailing of basic drug, it is customary to use an amine additive such as TEA, triethylamine, added to mobile phase A to mask or cover the residual cyanide group. That was widely used in the 1980 as proposed by Dennis Hill of UConn, which developed an HPLC UV method for Greyhound urine sample from dog tracks using TEA additive in the mobile phase. He found that the practice also enhanced batch-to-batch reproducibility. Here is the reference published in 1987. A more fundamental solution to the big problem was proposed by Jack Kirkland using high purity silica. Prior to 1980, a lot of the reverse phase column were packed with bonded phase from what we call a type A silica, which were loaded with metallic oxide. Metallic oxide tend to activate the residual cyanide group to make them very, very acidic. Research conducted by Jack Kirkland et al. And co-worker showed that bonded phase deriving from the use of high purity synthetic silica with very low metallic content, typically less than 50 ppm, could reduce residual cyanophilic activity and also improve batch-to-batch column reproducibility. Nevertheless, a buffer of high ionic strength is still required for those drugs or basic analytes which are very highly basic, such as those containing more than perhaps 2 amino groups, 2 ionizable nitrogen groups. This slide shows different type of residual cyanide group. Associated cyanide group are typically much less acidic. They are quite okay. And then you have increasing acidity going to the germinal cyanol and also free cyanols. However, the most objectionable type of cyanide group is those which are adjacent to metallic oxide. They're the worst culprit by activating the acidity of the cyano group because these metallic oxide containing empty d orbitals, which can allow the decolonization of the ions from the oxygens in the cyano group, making this proton highly protonated, very easily ionized. So surface groups, particularly aluminum and ions in the deep orbital metal, can activate cyano group to make them very acidic. Here's a number of references: Snyder Kirkland's John Dolan's book, this textbook, HPLC and UHPLC for Practically Scientists, also the YouTube channel of the various topics. I'd like to mention next video episode contains why don't we always need buffer in mobile phase A to eliminate peak tailing of highly basic drugs. Why do we use UV detectors, which is almost mandatory for regulated testing in QC? And why is acetonitrile the preferred mobile phases B in HPLC? This is the end of episode number 3. Why do we use acidic mobile phase A in pharmaceutical analysis? We welcomed LCGC magazine as our channel collaborator, and please subscribe to later videos.